Well, hello everybody. It is Tuesday night. We are live here on the show, and I'm actually using a couple of my apps to show you my numbers. So this is my Dexcom at 79. I have been fasting for a while, since Sunday. And then I'm going to show you, because I think it might be one of the final times that I have a continuous ketone meter in that is showing you a ketone of 1.9. So 1.9 ketones, glucose was 79. If somebody wants to do that Dr. Boz ratio for me. I am really excited about tonight's show that I have been putting this off. I have been wrong and I have seen lots of you out there and I have personal friends out there who've been telling me I was wrong, and I would much rather avoid this conversation, not do this at all. But you were right. So, I mean, there are some of you that I swear on every single one of the videos that I put out, you have put these comments about this topic we're going to talk about. I mean, I can be talking about, vegan versus, why vegetables don't matter. And you're still asking me questions in the comments for like 2 years. Every single video you've done it. Yes, I've noticed. Yes, I've avoided it, but I cannot avoid this any longer. I am going to talk about something that, I don't know, I'm very disheartened as I've done this. So we're going to get right into the topic. We're going to come back and show you some other exciting things that we are announcing this week. But let's just do the part that's hard for me. Let's go to the topic. Let me go to no, not that one. Hold on. There you go. This is one of them. I want to do this one. Oh, that's what I want. All right, let me take this down a little bit and make sure you can read what this says. This is a paper that came out just a few short weeks ago that I heard folks chattering about while I was doing the 21-day, but there wasn't quite a spare moment in all of the 21 days, this class that I do twice a year. And as I've looked at the report on what they were chirping about, I couldn't focus on it. I couldn't believe that this was true, or as true as it is. We've put the link of this in the show notes, so be sure to click on this because I don't know how long this is going to be up. I want you to point out a couple things here that I did not ignore, that this article was the review for this article. It is a peer-reviewed article, and it ties into a few questions that came right front and center as we were having our class over the last 3 weeks. But I want you to take notice. This review began in November of 2023. It just finished like the week that I did that I started this course. So I hadn't looked at it until just recently. But I want to point out a couple of things that you may not appreciate, but I do. That there are lots of crazy things that are said out there in medicine that physicians specifically ignore, and I have ignored them. That is really easy to do when we look for some stamps of approval from our advanced peers who help us to sort what's real, what's not real, what's fake news, and what's not fake news. And when somebody says that an article is peer-reviewed, and yeah, I'm sure there's shenanigans that are played in every part of medicine, but what peer review is supposed to mean is that an author puts together, in this case, a review of the data, submits his review of the data, and these really smart physicians, or doctors, PhDs, statisticians, advanced scientists, they're the cross-check. They're the fact checkers of medical articles. So in a peer-reviewed article, this one has several authors, of which I'd like to point out that Nathan Mead is one of them, Peter McCullough is one of them, and you have several others over here that are the authors. That is not who peer reviews them. You have about a dozen other peer-reviewed or peers who review the article, and that alone is shocking for this article. I'm saying this at the beginning of the article because if you're like me, what I'm about to tell you, you're gonna not want to. I don't want to believe this. I'm so irritated by this article and what it is showing me that that I had to put it on a live. I think this is a difficult thing for me to talk about. This is a very heavy article. There's no chance you're all going to read this. I'm going to point out a few things that took my attention and made me sleepless. But I want you to see that this isn't just a random person's opinion. This is a peer-reviewed article where in a peer-reviewed article, the authors or the peers will comment on the text, which means one of the authors, either the lead author or the co-authors, have to respond to the concerns and either document why that is true or not true, or they have to alter the text. And you can't publish it. That's why the review started in November. It did not end for 2.5 months because these peers, and there was like a dozen of them for this this article, cross-checked the information that we're about to go through here. All right, so we're not going to go through all 40 pages of this, but we are going to talk about some of them so that you can see what happened to my heart and what led to the reason I'll focus on this. So let's start by saying this peer-reviewed article is showing that that the COVID-19 vaccinations, well, they were a big deal over the last 4 years, and we did some incredible partnerships with the policymakers of our community and the scientists of our community to break some of the rules in order to serve the public in a time of crisis. So one of those rules that we broke was this right here, the Emergency Use Act. So the EUA included, hey, we need to do this right now because of what's going on in our community. But that comes with a risk that I knew about, but it probably isn't until this paper that I really appreciated that there were a whole bunch of things called significant adverse effects, that's SEAs, that included things like death and cancer and cardiovascular and autoimmune problems and neurologic disorders that were happening at a rate that we thought was acceptable because of the situation. All right, so I'm going to go through this and point out the things that bothered the heck out of me. So for the most part, you all know that when we look at this process, most of the time when you look for rapid authorization of a vaccine, the fastest we can do this is 4 years. And I can remember thinking at the beginning of the pandemic that there's no way they're going to get this done in 6 months. And you had all kinds of leadership people trying to say, yes, we can. And you're like, I don't know if you can make a flu shot. I mean, just make a flu shot in 6 months, let alone, get one that's for a virus that we had no idea about. That the trial period takes an average of 10 years to say, well, how dangerous is what we've been doing? So those are some of the basics to compare to. The first part of the article goes through a few things that give some examples. I like this one. Some examples include in 1955, a contamination of a polio virus. It had instances of Guillain-Barré syndrome observed in the flu vaccination in 1976, and there was a connection between narcolepsy with some of the flu vaccinations in 1999. We know there are adverse effects that come from vaccinations, and so we're not saying this never happens. We're saying that we thought this was an appropriate risk for the pandemic that we were living through. And I think if you're like me, when you get to the end of this article, it makes you think, what did we say yes to? What really did we say yes to? And I am not a conspiracy theorist. I really do believe in the hearts of people that are doing their job as scientists and doing their best to make the best policies and rules for a community. But the one thing that I think should have been pointed out first is, where do they talk about that they started using the word vaccination when they probably shouldn't have been doing that. Yeah, that goes through a few rules here. It also says that there were some bias. Oh yeah, here we go. There were some results that were strongly biased in order for it to work out. And never have I seen bias play out like this at this degree. All right, so this was the other part that bothered me. It bothered me from the beginning, but it really makes me nervous once you see what this. I had heard whisperings about this, but I didn't believe it. I did not believe it. So this bothered me from the beginning, though, that they kept using the word vaccine when it was not like a normal vaccine. This is a messenger RNA. This is a strand of single DNA or RNA that you make DNA from, so that you're teaching the body about how to respond to this by impacting your DNA. So it shouldn't have been called vaccines. It should have been called, let me put a blue in here. It should have been called gene therapy product, because it wasn't really changing the gene therapy. Let me just show you this slide here, where this is DNA on the top, which is double-stranded. Your body will make a messenger RNA from that, at which point you can make a protein. So maybe a spike protein. We'll get to that in a second. All right, we're going to come back to this in a minute, that other picture in a minute. Don't look at that. Don't look at that. There we go. Look at this. All right, so that the first of all, the terminology that they go into here says, yeah, why are you using those terms? There's another really interesting thing that you should be aware of, and that is they put this synthetic messenger RNA into a protective lipid nanoparticle. What that means is that lipid-based nanoparticle is going to be able to immerse into cell membranes much differently than anything we've ever used before. All right, so there's that whole thing. And now, this big circle here where I said, don't forget to say this. The problem with what they were playing with is there is no off switch for this. In a vaccination, your immunity rises and then it falls, and your body's done. With what they were playing with, there's no mechanism built into our bodies that has the ability to turn it off. Like if you take that DNA, you have a messenger RNA, now you're making a protein. Well, how do you stop making a protein once this turns it on? And that wasn't, I know. Somebody just said, oh, you're going to get canceled. I don't think so. I think I'm reviewing this scientific article, and I know I haven't done a lot of this, but there is, this needs discussed. This is a really important part of what happens as physicians when you trust that the scientific process is being used. And when they need to make exceptions to it, well, it shouldn't be that the exceptions mean break every stinking rule out there. All right, let me go through. So this section, and again, download this article, take a look at this. It's written, yeah, it's a little scientific, but it's not that scientific. So when S proteins enter the bloodstream and disseminates systemically, it may be contributing to factors that cause these adverse effects. And when we look at what the narrative is for what the research team was looking for, they really kind of, well, they did some things that you shouldn't do in scientific reviews or scientific processes. All right, so let's just back up. So registration for the trials, a couple things that I think were kind of weird. So I'm gonna read this part right here, because the study designed for the pivotal trials led to these, the Emergency Use Act, were never intended to determine whether or not a messenger RNA inoculation could prevent severe disease or premature death. It was mainly due to insufficient statistical power for assessing these outcomes. The power calculations was based solely on the reduction of COVID-19 symptoms, which was the primary outcome. And this limitation stemmed from recruiting these young, healthy people in what they were studying, and then extrapolating that to the data for elder people. All right, so let's see. Yeah, so yeah, I'll just, for instance, Pfizer's trial recorded one instance of severe COVID-19 death, whereas the Moderna's trial reported none, leading to a company to proclaim 100% efficacy for preventing severe illness. And I point all that out to say, yep, they took this healthy population of people, the ones that aren't going to have severe problems, they gave them vaccinations. One had a death, one didn't, but they said, hey, we prevented severe symptoms of COVID-19 with this process. All All right, so this is where I start to get really ticked off. You can see my writing gets a little bit bigger. I start putting exclamation points everywhere because this is where you start to see they played some really naughty games with the numbers here. So I want you to point out that they are looking at looking for the efficacy of mRNA product out of 162 from the 22,000 placebo recipients contracting COVID-19 and compared it to only 8 of the 22,000 vaccinated recipients. None of the 162 placebo recipients who contracted COVID-19 died. These numbers are too small to draw meaningful, pragmatic, and broad sweeping conclusions. Moreover, this is the part that's shocking. 170 PCR confirmed cases diverts the attention from the other finding that a much larger number of cases identified during the study fell under the category of suspected COVID-19. So they put them there and said, yeah, these people we think had suspected disease. 170 of them were confirmed. I mean, for heaven's sakes, confirm that they have the dang disease before we start making rules for the whole population on what was happening. They had 3,400 of them who they said, yeah, we think they had COVID. This acts like COVID. Well, run the dang test. Run the PCR. Why would you skip that step? Because what happens next is where these, so suspected individuals where, I'll just take this eraser so you can see where I'm at, and I'll re-underline it. Where the individuals exhibited symptomatic COVID-19, but they lacked a positive PCR. These tests were used in the trials where they were widely accepted. These were the standard of care for saying, do they actually have COVID-19 for some of the conclusions that we made? A total of 3,400 cases of suspected but unconfirmed COVID-19 were identified. This is a 20-fold increase between the suspected and the confirmed cases. And then if you, oopsie, look that I'm trying to see this actually, where there were 1,500 cases for the vaccinated and 1,800 for the placebo, but when factoring in both, confirmed that the suspected cases, confirmed and suspected cases, the vaccine efficacy against developing symptoms drops to only 19% when you start to say, hey, how well did we do at understanding did this vaccination reduce the symptoms? I remember reading this stuff, not knowing that this part in the background was based on not confirmed vaccinations. They suspected that it was COVID. They didn't confirm that that's what they were talking about. Okay, so it might be minor to you guys, but again, that's the first place where my trust got a little sideways. Okay, so the trial conclusion was predicted on a mere 100 of such COVID-19 cases, recording. Recorded in the placebo group. Let me go down to the next part. The lack of the ability to evaluate the severe illness in the trials reflected the real-world context, namely that the likelihood of severe COVID-19 hospitalization and dying from the infection has always been very low, but stratifying by age and infection fatality rate, the IFR, in 2021 showed an age-related gradient and an approximately 3 to 4 time increase in each decade. Okay, so, and as that went on, we did continue to confirm that. But what they didn't tell us was this next part. So we have overall all-cause mortality. And what did the pivotal trials reveal about the all-cause mortality? After carefully analyzing this, they found that 31 in the vaccinated and 30 in the placebo had a mortality relative risk of 1.03 compared to comparing the vaccinated to the placebo. Oopsie, I put that in the wrong spot. Comparing the vaccinated to the placebo. These findings became interpreted as no significant difference or no gold standard evidence showing that these mRNA vaccinations reduced mortality. Huh? The lack of significant difference in the deaths between the study arm is noteworthy. The true mortality impact remains unknown in this context, and this fact alone is relevant. That a good trial evidence of reducing mortality than to take a vaccine where the trial evidence does not show convincing evidence to improve the survival. And then we get into, so this keeps going on, where what's really being talked about here is that not only were they looking at when the death rates, were the symptoms of the COVID-19 measured fairly to impact, is it worth the risk of a vaccination that isn't being that isn't new, that is new to us? Not only is it the first mRNA vaccination that I'm aware of, it is massively delivered to all our children, pregnant women, and elders. This whole section in here goes through that they're playing funny games with the math. One of the best parts about statistics is when people start to play with the math, you might not be able to see the exact place that they played with the math, but it doesn't plot out in the expected way that the statistics say it should. And here's the part that I wanted to say. Both deaths are counted in each arm of the trial. This is really important. I know that this might there's two more points that I need to make, this one and the next one, which really shocked me. Okay, every 6 months, let's see, where did it begin? Okay, so they extended the portion of the trial, included 4 months of an unblinded period, where most placebo participants crossed over to the vaccination group. Okay, so here's another thing that they did. When you look at a trial of people, you have a placebo arm and you have a treatment arm. So what they did, and what I'm about to point out here, which was totally naughty, is they put them in the trial arm, they put them in the treated arm, and then they let them continue for, I think it was a short period of time. I want to say it was like, 4 weeks, not even 4 weeks, where if you got the placebo version of it, and you got so far out, you could say, yep, they told them you had placebo, and then they could change their mind and go over to the vaccinated side. And now what they were doing is they were using some of the people who started in the placebo line but ended up with the vaccination in the data for the placebo. So they got the vaccination, they had a significant adverse effect, but they're being counted in the people who didn't get the vaccination because that's where they started. They didn't fix that. I mean, first of all, they should have never offered that to them that early in the process. And I know that all rules were off when we look at the way this turned out. But let me just read this because it's heartbreaking. Oh, I want to do the other one. I think it reads a little easier. Not that one. Dang, I did it again. This one, yeah. All right, so they extended the portion of the trial. Okay, it was 4 months, not 4 weeks, in the unblinded period, in which most placebo patients crossed over to the vaccination group. So they went from being placebo, and now they're in the vaccination group. During this phase, there were 5 additional deaths, including 3 in the original vaccination group, and 2 among the placebo participants who chose to get vaccinated after 4 months had gone by. When these 5 deaths are included in the vaccination deaths, the total comes to 20 deaths in the vaccination group and only 14 in the placebo group, which would represent a 43% increase in deaths when you got vaccinated, which is not statistically significant due to the small count. But in the FDA documents, a total of 38 deaths were reported, with 21 in the vaccination group and 17 in the placebo group, representing a 23% increase in all-cause deaths among those who received 2 doses of the primary series. This suggests that the 2 placebo participants who died after getting a messenger RNA vaccination were counted twice. They were counted in the vaccination group and they were counted in the placebo group. And then they talk about how they would run those numbers. So you don't have to be a statistician to understand what I'm trying to say, which is they played games to have this bias towards the favorability of the vaccination. Now just wait, hang in there. I know you might not like numbers, but this is really an important process. And if they take this paper down, I want to be part of the people who said there was more to this than they ever told us. Okay, so this is important to me. Number needed to vaccinate. So when I look at there's lots of shortcomings in this. This is an emergency process, but let's get to the brass tacks and say, well, how many people was I going to need to give this vaccination to in order to get a saved life, somebody who didn't die? So let me see where it goes in and it talks about this. It tells you about that. Okay. Okay, here we go. So the approximately 50, I'm going to use a different color because I'm running out of scribbles, 52,000 vaccinations would be needed in order to prevent one COVID-19 death. Now, they go through talking about all this, how this number needed to vaccinate, they are taking a conservative approach to this. So as you do the math, they're rounding in the way to be conservative, giving the benefit of the doubt. And it's saying this exact injection. It is estimated that about 2 lives could be saved for every 100,000 injections. And then they go to say, but now we see that we have 27 deaths per 100,000 in the same batch of injections that were happening because of adverse events. So you're going to save 2 lives, but you are going to have 27 deaths as a result of the significant adverse effects. And yet this was not reported. There were nearly 14 times more deaths caused by the modified mRNA injections, and it shows you that in Appendix 2. Okay, so one last thing, and then I'm going to go to a picture, and then we can take your questions, because I think this is worthy of talking about. It's something that I have made fun of my good friend Colonel Auk, who kept telling me, there's more to this than you know, there's more to this than you know. And he worked for the military for 50 years. He's also a physician. And he kept saying, don't do it, don't do it. I'm like, don't tell me this, Colonel. Come on, they'll never be that evil. They would never be that evil. So I highly recommend reading through some of the stuff that I'm scrolling through right now. But I want to get to the final one, the one that this is where they played funny games about when they stopped counting and they held off reporting the death dates until after they stopped counting. So you see in here a place where they said, yeah, okay, I'll tell you this and then I'll read it because you'll think it's not true. They didn't report the date that the person died. If there was an adverse effect, there's a death date. That's not a difficult thing to follow. But they reported what date was the death reported to us. And so it delayed the onset of the death in the vaccinated people so that it didn't look like it was as deadly. Let me just show you this. Okay, so I'll just underline as I do this here because it's easy for you to follow along. Okay, so let's go to here and say Pfizer used the dates of the death was recorded in subjects' case forms, which they maintain, including the death or hospitalization within 24-hour window, a guideline likely followed by the trial staff. I still didn't get it. Let's try one more time. Okay. The researchers were able to access the narrative reports on a few critical subjects that provided explicit notification that the subject's date of death was prior to November 14th, which was this cutoff date for the data for death rates associated with the vaccination. They required immediate reporting of significant adverse effects, including death, blah, 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 blah. Nevertheless, Pfizer used the dates that the death was recorded in the subject's case report form. Like, it wasn't the actual death date, it was delayed. These delayed were greatest in the vaccination subjects prior to that November 14th reporting time. Pfizer had Pfizer used the actual death dates, 2 additional vaccinated subjects would have been included in this emergency use application or act application. This discrepancy was crucial. It changed how the outcomes were, especially when you realize that 2 of these deaths were cardiac based. Twice as many cardiac deaths occurred before. Yeah, they played some funny games, is what they're saying in this stuff here. They played plenty of funny games with the math. Okay, now the last thing I'm going to show you, which I really still can't believe Yeah, this is one of those places where they say, yeah, they recommended it in pregnancy when they knew that absolutely not one single subject in the trial was pregnant. I think that's just egregious. But let me go to this final part where then I'll quit chirping and I'll take your questions. It's not there. It's a little bit further down. Oh, yes. Okay, quality control issues. Okay, so first of all, it's a trial that this should not be an issue. So given the novelty of this mRNA technology, it was prudent to establish the regulation and the quality of the long-term safety for this monitoring. So they go to a key issue that that could help explain why individuals succumb or die while others do not is the vaccination type and batch variability. I'm like, well, why would the batches vary so much? Isn't it just an MR? I mean, there's the standards to produce these. Okay, due to the inherent instability of mRNA technology, some batches may contain extremely low levels of the messenger RNA. Some batches were contaminated with double-stranded RNA, as documented by I can't remember what EMA stood for both the Madeira and or Pfizer and Moderna products. The double-stranded DNA has a high potential to trigger immune inflammatory reactions such as myocarditis. So here it gets a little bit, this is where it really bothers me. So you say, well, I don't know much about these vaccinations. Is that like a normal thing to find in your vaccination with an mRNA? Is you're going to have some double-stranded things? They say each batch variability further complicated in recent findings of DNA contamination. Again, DNA contamination. First of all, what the hell is what is DNA doing in a vaccination that is supposed to be for the protein production to fight off this virus? But in the analysis of multiple vials, high levels of DNA contamination in both a monovalent and bivalent vaccines, in orders of magnitude higher than the limitations. Oh, I know. I think this is the regulatory board for Europe, and FDA is what we call it. So it was magnitudes higher. I mean, 10 nanograms per dose is what's allowed in our FDA, whatever, it's 330 nanograms per milliliter there. But they said it is worth noting that it just says somewhere right in here where, oh yeah, here we go. The FDA, oh yeah, it's in yellow. I put it down here. So what they found was an enormous amount of DNA particles in the RNA that I injected into me, into my kids, into my patients. So how does such dangerous, large-scale contamination escape the scrutiny of the public health officials? And were the manufacturers aware of all of this excess DNA that was found inside these vaccinations? Oh yeah, this is the part that I wanted to say. So when you have foreign DNA in your human genome, it disrupts the existing natural genetic sequence. It also carries a further risk of disease, including cancer. I mean, DNA in your body, very dangerous. That's like CRISPR, except it's random chance of what the heck's in there, because it's a contaminant. It's not intended to be in there. What's it doing in there? So they said, well, did they know? It is important to note that the process-related impurities were absent from COVID-19 products when they applied to the government to get all these exemptions. So when they went to the registration trials, there were no impurities. It was strictly the mRNA. What I have in my mind that I imagined I was giving to people that I was receiving, virtually all doses used in those trials originated from clinical batches produced using what was known to be process 1. But as they went to say, well, now we need to use a different process to mass produce this mRNA, which is called process 2, and that seems to have, utilizing bacterial plasmid DNA, the process 2 alterations included modifications to the DNA template employed by the RNA transcription and changes in the purification phase and adjustments in the manufacturing process for these lipophilic processes. They go on to talk about how much was in them and how this was not, huge potential to be harmful. There is an 803 increase in autoimmune disorders when people had these vaccinations. And it goes on to say, we not only know that, we know that as you look at some of the images from people who have autopsies from death due to myocarditis, I wonder if this is, here we go. This spike proteins are found in the cells of all human heart cells with spike proteins inside the muscles of these patients. Again, spike proteins are not supposed to be inside us at all. The people who had COVID didn't have spike proteins found in their cardiac tissues. The people who had the vaccinations, the ones who had this messenger RNA that made a protein, and then they made the protein and didn't have the off button, well, they're now found at autopsy in the cells of cardiac tissues. And here are that sounds ridiculous. It sounds like something I wouldn't even believe until you start looking at the autopsies and wondering, hey, what part in this did I play? And first of all, how do you say, I'm sorry to all of you that have been putting comments in my videos for like a year. Take a look at this, take a look at this. And I said, okay, they're just chirping again. And my good friend Colonel Auk, who told me from the very beginning, be careful, look what's happening. This is sneaky, why are they holding this back? And he questioned everything. Now he questions everything, so it's like the boy who cries wolf, that I should have been paying attention when he cried wolf this time, but shame on me when there were places that this information was more readily available. And let me just close with this story, that when I was when we were in the 21 day, there was somebody who asked, should I take the vaccination for, and then they filled in the blank. I think it was, they were talking about the flu vaccination, and they were also talking about, shingles vaccination. And the paranoia, the lack of trust of so many people in that class that said, I don't know, I don't trust, I don't think we should have any vaccinations anymore. And I got irritated saying, why? Again, I had not read this report, but in fairness, this report talks about a distrust that happens when your people who are supposed to be dedicated to a scientific process, to standard rules for studying people and subjects that say, I volunteer to be in front of the line for this shot, and I will take the risk. And when you disobey, when you dishonor that trust, when you betray that trust, now they're saying, well, can I trust these other vaccinations? And I'm like, well, of course you can. And it's hard for me to say that without just saying, oh no, look at how long is it going to take before the world would trust, that I will trust what they're telling me. And then what was my part in it? How could have I been a little more, again, I had all of my kids vaccinated. I had all of the people I loved vaccinated. And when you look back and say, dang, look at what it did to especially the kids. Anyway, I say that to you to say, this information is out there. I don't know how long this report will be there. I want to do my part in at least spreading the information that's in this report, because holy mackerel, did a lot of work go into that report. And if you're trying to find confidence that there are good people out there trying to find the truth, to live in a time of scientific transparency, and I think the ketogenic diet is part of that, where as I've walked through saying, here's what I'm seeing clinically, here's what the research is telling me, and I'm trying to pair those up as quickly as I can, with reserving the right to say, oh no, I was wrong about that. And when it comes to a place where I was wrong, I don't know that I get much worse than telling all the patients that I said, get the vaccination, get the vaccination, get the vaccination. All right, so let me go on to something much easier to talk about, which is not my continued failures in life, but to say there's a couple things I wanted to point out. Several of you have asked where to get the continuous glucose monitor. You do need a prescription for that. We have made a pretty easy process for a continued glucose monitor prescription. If you click on that link, it leads you to the Meaningful Medicine partnership that we use to help with that. I'm going to scroll down to the bottom here and say that Levels is another place that you can get a prescription for that. They have a subscription, so you pay for their continued support and education. They also offer you both types of continuous glucose monitor, where I just offer the Dexcom version. I also have put out an ask and was really pleased to see some of you click on this, that when I look back at some of my dreams, one of them is to help people who are out there taking care of mental health issues better assess brains at the beginning of a story. So I've partnered with some jails, I've partnered with some therapists and some therapy teams on what is it that I do when I'm assessing somebody's brain. And that assessment is something that's asynchronous, that I do this for anybody who takes the brains course, but I'm looking for other people who could help me find a place where assessments need to be done on a higher level, or at least on a more thorough, without as many, like inside a jail. The problem is getting the assessments done inside a jail is difficult. Let the iPad go into the jail, let my counselors be on the outside of the jail. Anyway, so I still want folks to be aware that that is a huge dream of mine, and I've been a little shy at sharing that with people. But it's, yeah, if I could put one thing on my epitaph, I hope that assessment is in the use before I die. I mean, massively in the use. All right, there are several other things people wanted to know about. Somebody asked me, what is the deal with your favorites page? This is like the commercial for the work that I do, the service work that I do. These links and these promo codes do give us a little commission. I don't put anything on here that I don't use or somebody in my family doesn't use. And we have some pretty fun things we're going to teach about over the next few months that are on this page as well. So I encourage you to go there. It's just one way you can support us. I also wanted to let you know that we do have that a product on sale that I talked about last week, but right now it's stuck between our warehouse and Amazon. So tune in next week on that sale. Hopefully we can find, there's still a few hundred of them for sale, but you can't find them anywhere, so don't go clicking yet. Instead, I guess you can click on any of the other stuff that's for sale in there. All right, so I'm going to go over to the questions, and let's go back to here and say, I do look at your questions. There are several of you on tonight that are a major part of helping me put on that course that we just got done with, and I was just starting today to read some of the reviews that, oh my gosh, they make me smile. They really make me smile. Let's go here. So let me take that down just a little bit. Didn't fit as well as I thought it would. Okay, so Karen writes in and said, I'm concerned that leaving a ketone drink out of the fridge could be a problem. Can you tell me if they go bad? No, they don't. In fact, one of the things I have to do is check shelf stability. And the one that's the newest one that I had to just get approval from on the shelf was the Pucker Up. And it sat out on the counter for 4.5 years at room temperature and was still good. The salts are even more stable. So when you put the salts into water, which is what the powders are, they're actually a salt, they don't go bad. They are a ketone, as is a ketone, so they're not going to go bad. It's a very good question. I've never had that question before. Neil writes in and said, how will being in keto affect those who had the first and second round? You know, it's a really tough question. That's why I went into this, Neil. Thank you for asking that. He's asking, how do those of us that had those first rounds of vaccinations, how will it affect us? And, what led me down the rabbit hole of even, getting close to this paper was what happens with that chronic inflammatory state of cytokine storms? And what can you do to still have the cytokines or the inflammatory response that heals a body, but that does not constantly just, simmer or brew an inflammatory response that kills off tissue, that hurts you. So how do you have enough response to help your body, to protect your body, without the flood that happens in many of the patients with chronic diseases like high insulin and diabetes? So I had this question also this morning at my support group, that when people go into a ketogenic state, and there was a gal who's been doing consistently keto for a few years, she's doing great, but she says, I did that 72-hour fast and the scale dropped. And I said, well, what were your ketones at the beginning of the fast? And she says, oh, they were like, 1. They were good. And what were they at the end? And they were like 3 or 4. They were pretty good. So she had a good surge of ketones during the time of her fast. And I said, well, what you can guarantee when you have a high state of ketones is that you are pushing the anti-inflammatory. You are wringing out the inflammation the best that it can be. The other thing that you're doing is that there is a signaling agent that when your body sees the signal of ketones in circulation, it protects it from breaking down muscle. So that the weight she lost during a surge of her ketones was very likely to be mostly water and fat and not muscle. Well, how does that answer the questions for who had the shot? When you look at the inflammatory response that people have for a messenger RNA, for proteins being made in their body that they shouldn't be, that the autoimmune, that immune response that is erroneous, that's attacking, well, whatever we injected in it, but also attacking ourselves, the way that gets reined in, again, something I did not learn in medical school. I mean, we knew that if they slept better, their autoimmune problems were better. We knew that if they were under less stress, that their autoimmune problems were better. But never did I read that you could reverse an autoimmune problem by really reining in the extra abundance of an immune system going in the wrong direction, which to me is what a cytokine storm, what a chronic inflammatory response is happening after those infections or after those injections. So the effect of an advanced ketogenic state is you get a high signaling agent to say, remove inflammation, and you get to protect that you're not breaking down muscle for energy. But Neil, or maybe it's actually Nell, sorry. Nell, I'm right with you. I have no idea what that really means, and it bothers me profoundly. All right, Brenda writes in and says, so do the same people make Paxlovid, and does it have the same problems? I don't know, actually. Honestly, one of the parts, Brenda, that I'm going to take a little piece in, and maybe it'll help you, is that when you look at why the government was able to do the things they were doing, it was because they were under that Emergency Use Act. I don't think Paxlovid is under the Emergency Use Act anymore. I think those are all like, we're out of the crisis, people go back to normal. And in the normal process, it really does protect you. There are checks and balances, and there are really, not just good people, but healthy attention being paid to, did you make a mistake? And I think that's the beauty of a peer-reviewed article, that there's a bunch of articles out there that I don't waste my time looking at. I spent a lot of time looking at this article, diving into the extra articles behind the article and the process check, because, well, I didn't want to believe it, but it should be believed. There are lots of checks and balances, and normally in places where you're having the Paxlovid, which I assume is an autoimmune disorder one, it's got the crosses, checks and balance in it. All right, so JFR says, can fasting help change the change in our DNA that was made? Yeah, our fasting can get rid of proteins we are now making. So that's part of what I'm hanging my hat on. I don't have any evidence for that, but when you look at what is the strongest signal when people are at that advanced ketogenic state, like the gal was talking about in the meeting this morning, and that is that our cleanest DNA is being replicated. The one that is making an error has inflammation in it, and that inflammatory process promotes the mistake in an autoimmune disorder. At least that's one of the theories that seems really to to really match up lock and step with what I see clinically. Why does this autoimmune disorder reverse when they stay in a ketogenic state, weeks after weeks after weeks? And now 2 years later, they say, I don't have that problem anymore. I mean, I have a couple of young people that are in high school with an autoimmune disorder that's starting. And I just want to grab their parents and say, turn off the inflammation. What does that mean? That means do not feed them the processed foods that are everywhere in their life and that are really hard to get rid of. But for your behavior, they'll be copying you. So put you in a ketogenic state and watch what happens to the choices of your children. So JFR, I want to believe that it can do that. I don't know if I'm right or not, though. Bill said, we are 78 in good health. Should we continue to get COVID shots? Boy, now that might get me canceled. Well, I'll tell you, if I was 78, I wouldn't get it. I would say, no, it's not. I mean, what they're telling you it's doing and what it's doing, dang, it doesn't match. I mean, that's what that whole report is saying, is that they broke some rules. They made some assumptions, and then the assumptions turn out not to be true. And there's a whole bunch of funny business in the numbers. I see Margaret Henry writes in, is that report available to the public? Yes, it's in the show notes. Please click it. Please download it before. I mean, I don't know. I'm not paranoid, but I cannot believe the evidence that's in that report. So Sue Monroe from Iowa, nice to hear from you. I have had all 4 vaccinations and COVID 3 times. I was diagnosed with a heart condition last year, enlarged aorta and root. Is it related to this? I don't know. I mean, let me just show you this. So these biopsies are in people who have, that is a spike protein in each one of them. That cardiac tissue is not supposed to have a spike protein anywhere near it. That red is a spike proteins. And again, when they had just the vaccination, they don't have this incredible production of a protein that doesn't belong in our cells, let alone inside the tissue of a cardiac cell. And so, do I think they're related? I don't know. I don't know, Sue. I'm sorry. I wish. Yeah, I'm sharing this with you in a place that's very vulnerable, because I can't believe that it is as true as it is. Ellen writes in and says, so what is the source of the external DNA? Yeah, it's not horse. If you go into the report, I want to say, I looked this up because I think it was, what's the word for monkey that begins with an S, like Sabian? I had to look it up because I'm like, why do they mention this here? So the external DNA, I think it was in the mass production of it, but it sure it's supposed to have 0 in there. It's got billions of particles. Again, what got approved by the FDA, by the committee, was something that had no DNA particles in it. But you go take a vaccination now, they look at it, and it's got the snippets of double strands DNA, not messenger RNA, double strands DNA. Anyway, it's very frustrating. It's very shocking. Yeah, anyway, last question, and then I will say, sapien. Thank you Marvin, that's exactly the word I was looking. It was sapien, it has sapien DNA. Well, the word sapien is Latin for like ape or monkey, yeah, Homo sapien versus sapien. Yeah, that's the word I was looking for. Anyway, reading it is a little disheartening. You might want to be drinking ketones when you do it. Last question, Debbie writes in, how long does it take for your LDL to go down? I've always had high LDL and low lipids. They always want me to take statins because my LDL makes, my HDL makes it higher than, 200. So when I tell people what to do on the ketogenic diet, I tell them you want to be stable in a plan for 6 months. That when you're looking at LDL numbers, what you're really trying to assess is you're moving energy around a human body. How well you're doing that, how consistently you're doing that, is something that stabilizes once you get your head around how to be on a ketogenic diet for real. Not just a flash, I'm going to lose weight for a wedding, not just I'm going to hurry up and, reduce the problem that is happening that you want to go away, but something that you're in a zone, heading in the right direction, and you're not going to turn left at Albuquerque next week. You're actually going to stay the course. So I would say 6 months of stable ketogenic lifestyle before you look at your cholesterol. That's what I do. All right, folks, I am going to sign off, improving your health one ketone at a time. We will see you next week, and I promise not to chirp about medical literature next week, but I would love feedback on, did you learn something, and are you going to share this?